Okay, so
Good afternoon, everyone.
present
ScienceBug
now.
welcomes you all for today's webinar on From Cell to Solution Mastering Protein Purification and Characterization by Dr. Sunny Singh. Before starting the session, it's a humble request to everyone to please keep your audio and video settings off in order to avoid disturbances during the session. And if you have any questions, you may use the chat box. All right. So now I would like to introduce you all to our guest speaker. Dr. Sunny Singh is a seasoned protein sciences expert with over 17 years of international experience in biotechnology, diagnostics, academia, and industrial research. Born in Mumbai, India, he earned his PhD in chemical biology from University of Sussex. Dr. Singh's career spans academic research at Technion in Israel, where he solved antimicrobial peptide structures, to leading COVID-19 molecular diagnostics in Mumbai during the pandemic, where he managed high throughput RT-qPCR workflows and trained over 50 staff. Today, he serves as a senior scientist at LenioBio GmbH in Aachen, Germany. He has contributed to global health and innovation through projects on therapeutic antibodies, vaccine antigens, and enzymes. Dr. Singh's achievements include winning the Brain Race Startup Challenge for proposing innovative glioblastoma treatment and supporting anti-doping efforts during London 2012 Olympic Games. Beyond science, Dr. Singh is passionate about education, community development, and environmental sustainability. He envisions a progressive India with skilled, well-educated, and economically independent citizens. His dedication in science and community makes him a notable figure in this field. With this, I would like to thank Dr. Singh for accepting my invitation and joining us for today's webinar. Over to you, Dr. Singh.
Thank you for the kind introduction. So I'm glad to be here and to meet you all. So how many of us are here right now? 30 of about 40 people, or how many more? I'm guessing just under 40, right? Okay. So hi everyone. So my name is Sunny. So I've been living in Germany, working in Germany from the past 3.5 years now. So as already I have been introduced, so just I would like to give you some knowledge, share some knowledge that I have gained over the years, thanks to my supervisors during my PhD, my lecturer, Dr. Amrita Chima ma'am, who is a professor at Georgetown University in USA. So she was my inspiration to, take up biotechnology, as in after my bachelor's, for me to continue and keep going and pursue science, because I actually didn't know what to really do after my BSc in biotechnology. But here I am after so many years. So I guess you guys are doing microbiology, or are you also doing biotechnology? Because just wanted to know in general what my audience, how much should I explain the things that I'm going to talk about. But anyways, so I completed my biotechnology in 2005 from KC College in Churchgate, Mumbai. That's University of Mumbai. So that was like the first batch of biotechnology, and nobody knew what was there in the future for us. So we didn't have any mentors, we didn't have any support system. So it was all trying to find out things on your own, and somehow we just managed to, some of my friends went to the USA, and I went to the UK, and I stayed there for many years, master's, PhD, and then moved to Israel, and then came to Germany after India. So let's begin. So today I'm going to talk about protein purification and characterizing protein molecules. I mean, there are some things that I will tell you by uh, word, like audio, because I didn't really put too much information because it will be too much to take. But I can also explain you in between as I am talking. So please bear with me. If you don't find it on the slides, you can take notes as I am talking, and you can always contact me afterwards on LinkedIn. I will share my contact details, and you can contact me there for any questions or any collaborations, any ideas that you might be having for moving forward for your career, for your research, or maybe some of you might want to start up your own company. Maybe you might get some ideas after this webinar. So that would be interesting. So why not? So let's begin. So okay. So in this webinar, I will give you a sort of an introduction about, going to tell you about what I'm going to talk about. So the importance of protein sciences, what are proteins and how important are they? What are the different protein expression systems? And what are the protein purification principles? And what are the ways in which you have to go about doing protein purification? Because you know, doing protein purification or cloning, all of these require some skills which come from repetitive learning, handling pipettes, pipetting small microliter volumes. In the beginning, we don't get it quite right, but as time progresses, we become better at it. So I was also making many errors when I started my journey, and then later on it became better. So do not feel disheartened if things don't work out in the first instance. So there's always time to recover and correct yourself. So, and then I will also talk about protein characterization and analytics. What are the career options in protein sciences? And then we can have a Q&A session. So maybe in an hour's time, if it's too long for you to take so much information, we can have a 5-minute break just as a breather, so that you can take a bio break and grab some water or some snack, and then we can resume. So let's talk about proteins and their role. So as you know from your textbooks and all these years from studying, that proteins are also macromolecules like carbohydrates and fats. But proteins are all the more essential because they are fundamental building blocks, and they are functional molecules for all life forms on our planet Earth. So it is absolutely important. I mean, you can debate with viruses about viruses because viruses are not really life forms unless they enter. But still, there is a potential that they might come alive. So we don't want them to come alive because they really cause a lot of problems. But they also have a lot of protein, decorated protein on their surfaces, because you already know the SARS-CoV-2 virus, the lovely looking shiny decorated virus which caused so many problems for the COVID-19. But yeah, viruses are also made of proteins, and they come alive when they enter cells. So proteins overall perform a vast range of critical functions. So as we know, all our life processes right now, as we breathe, and so many times, we don't even think that we are breathing, but all of the life processes are ongoing. So all of the pathways that TCA cycle, tricarboxylic acid cycle, and all of the metabolic pathways are continuously ongoing, and this is all happening because of proteins. All of the enzymes that are in our body, they are helping you survive, live, and also autophagy, where you are recycling all of whatever you don't need, and the body is really making use of all of the resources that it builds, and it recycles. So they catalyze metabolic reactions. So so enzymes, so all of your ATPases, all of your different enzymes that are in the body, which are performing, like the liver enzymes, also like the AL, ALT, or In the earlier, we used to call them SGOT and SGPT, but now they have been renamed with new nomenclature. So the liver enzymes, the stomach enzymes, and all of the enzymes in our cellular system providing structure. So you have collagen, keratin, so hair, nails, transporting molecules. So hemoglobin transporting oxygen with the help of oxygen binding to the heme, and replicating DNA. So your DNA polymerizes. So your cell cycle regulation, all of the cell division, and all of the DNA that is required for each of those cells is done by DNA polymerases. So, and also responding to stimuli. Now, this is not really a protein, but this is a process. So, the process here, it involves the process of phosphorylation. So, phosphate molecules get added onto proteins. Whenever you learn a new skill, whenever you memorize something, your new neuronal connections are made. At the same time, the proteins in your brain, they get phosphorylated. So whenever phosphorylation and dephosphorylation is happening, it's about memory formation and memory recalling. So when you form a memory, you have phosphorylation happening. When you recall a memory, you have some dephosphorylation happening. So this is a very interesting phenomena where people do a lot of research on snails as model organism, and they try to, have you, do you know the bell experiment where the dog, they ring the bell and the dog starts salivating because they have trained the dog to give the dog food when the bell sounds. So as soon as they ring the bell, the dog salivates. So the same way, a friend of mine, he trained snails by putting sugar in the water, and the snail would think there is food. So what he would do, he would train the snails, and the snails would come close to the source, and then he would take the snails. Unfortunately, he would have to kill the snails, and he would take sections of their brain, and he would do microscopy, and he would do protein extraction from their brain. And what he saw, this was like about 10 years ago in Sussex University. What he saw, that you have phosphorylation happening as the snail is gathering, creating memory to remember that this is a stimuli and I need to go to the source. So that's interesting. And then immune defense, your antibodies. So all of the antibody in your body, like IgE, the allergic reaction antibody, or IgG, and IgD, IgM, all of these antibodies, these are your defense antibodies. Some have uh, what do you say, a constant role. Some have a dormant role, but they get activated later on. So these immunoglobulins are also proteins. And you already know with the immune therapy, even during COVID-19, some people were given the antibody treatment along with the, before the vaccines came out. So some people were given um, lifeline by putting antibodies for COVID-19 so that the body could prepare itself, because they didn't have the ability to prepare antibodies because they were weak. So this was also done. So now let's see the importance of protein sciences. Now, the crucial importance of protein sciences. So, I mean, you see, proteins are our cells, let's say the laborers or the workhorses. They drive metabolism, they do the DNA replication, they help in the signaling mechanism, they help transport, and they give structure, they give a framework. So, proteins are very important from the life systems point of view, and they also translate genetic blueprint into biological function. So, genotype to phenotype, you have read in your school books, and also when you started your junior college or 11th 12th, you saw how genotype, and then you'd have the Mendelian genetics, and then you have the phenotype. So, F1 F2 generation, you see some sort of genotype crossing is going on, and then the phenotype. So the plants appear different because they get, crossover. DNA crossover. So DNA, and always the expression will be in the form of protein. So that's the phenotype. So that's why protein expression. So whenever you do microarray based, what do you say, investigation. So maybe like many years ago when I was studying, that time microarray was a very hot topic. Now microarrays are fine, but there are even better technologies. So to look at which genes were turned on, so there was a microarray analysis at the time being done to see which genes are turned on. And after the human genome got sequenced, the role of microarrays kind of got, I wouldn't say diminished, but they really, we utilized it. We exploited the microarray technology to look at all of the genome of human, the whole human genome, to see what was on and what was off, and then you knew what was junk DNA, what was active DNA, and then yeah. So the phenotype is in the form of protein. Now, revolutionizing medicine and health. Now, you see so many diseases that come about, that happen because of malfunctioning or misfunctioning or misfolding of the proteins that are supposed to carry out their original function. They somehow do not perform well enough, or they don't perform at all, or they somehow are just not present. They somehow get deleted. So there is absence of these proteins, and that's when diseases happen. So, like for example, a diabetic, a person with diabetes. Why do you think? Can anyone tell me why do you think they test their HbA1c? What is HbA1c? Can anyone tell me? Can any of you speak, or you can't? No, Kanvi, not insulin resistance. Anyone else? What do you mean by HB? Yes, that's correct, Megha. Maham, that's also correct. Hemoglobin is the right answer. What is A1C? What does that tell you? So something hemoglobin, yes, diabetes. But what does it actually mean? What are you checking? Mm, no. So yes, you are checking the glucose level, but what are you actually doing? So yeah, that's fine. 3 months, but I wanted one answer that was. Have you heard of glycosylation of proteins? Glycosylated. Yeah. So what is glycosylation? Glycosylation is carbohydrates or glucose. Now, if protein is glycosylated, it has the addition of sugar. Yes, Mahadev. It's the addition of sugar. So when a diabetic has to get their HbA1c tested, it means it's the red blood cell hemoglobin which gets glycated. Yes. So glycated RBCs, because if the person has been consuming his diet and has not been controlling his blood sugar, what happens is the excessive sugar molecules that are floating around in the body, where would they go? They are circulating in the blood. Now, if there is not enough insulin, the pores of the cells don't open, so the glucose molecule just keeps circulating in the bloodstream, and the cells stay hungry and they are not able to open their pores because there is either not enough insulin in the body, or the insulin has lost its sensitivity, or cells have lost their sensitivity to get the insulin dock, and then the pore would open to let the glucose in. So what's happening? The RBCs, instead being in contact with the glucose, are getting more and more and more sugar, 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 sugar. So you have more of the RBCs, and the hemoglobin of the RBCs, which is the protein, is now getting glycated. Means you have more of the glycan. That's why when the doctors check the sugar in 3 months or 6 months, they see, okay, let's check your diet history. Let's see how naughty you were, that you were consuming sugar or you were not controlling your diet. That's when you see, oh, HbA1c is very high. It's 8.7 or 9. That's terrible, because ideally it should be 5.5 to 6. If it's a diabetic patient, if he or she is a diabetic patient, it should be about 6 or 6.5. Anything more than 7 is not good control. So I'm telling you this because, unfortunately, or let's say I was the lucky one, I also have diabetes, but I have diabetes type 1, juvenile diabetes. I have diabetes from a young age. I take insulin every day, many times a day, before my meals and before going to bed. So I take insulin. So that's why protein sciences also became very important to me, because without insulin, without a recombinant protein like insulin, I probably would not be speaking to you. So, thanks to biotechnology, thanks to the invention, 100 or more than 100 years ago in 1922, that we have insulin which can be injected. So, anyways, therapeutics, you also have insulin, vaccines, drug discovery. You have to identify the targets, which are protein targets, and the proteins have to be crystallized for their atomic structure. So, X ray, crystallography, cryo EM, and NMR are ways how you can get the atomic structure of protein, which is very important. Then you have therapeutics. I already told you, antibodies, insulin, vaccine, enzyme replacements, because some genetic disorders also need enzyme replacements, where certain enzymes are not produced, like Gaucher's disease, where your spleen and liver are badly affected. The fat gets deposited on your liver and spleen, and they stop functioning over time. And some kids die within a span of 2 years age. So right from a very young infancy to 2 years, they can't live any longer. So Gaucher's disease, you can Google it, you will know what I'm talking about. So diagnostics also, and then more importance is industrial enzymes. So now you see there's a lot of boom in the food industry. You have a lot of packaged foods. How do you think they package these foods, and how do you think all of these juices are made? So some of these enzymes are like pectinase enzyme. So when you have to process apple juice, you have apple with the peel. So the peel has pectin. Now pectin will make your fruit juice not so likable by you, because when you try to slurp it in by a straw, you will not enjoy it so much, because you will be like, oh, इसमें छिलका क्यों डाला, निकाल दो ना. But pectinase is the enzyme which actually gets rid of the pectin and makes the juices smooth and easy to sip. So this is an industrial, and then you also have biofuels. So bioethanol, you have heard. There are also enzymes that process the biomass to get to a stage where the carbohydrate is easily processable, because there is so much of the cellulose linkages that the cellulose has to be broken down, and then you get to process um, the biomass very effectively. So, how do animal based protein sources compare to plant based sources in terms of diabetes? Well, diabetes management, I mean, we can talk about that later, but I would say plant based sources also proteins. So, the ideal thing would be to have more protein, less carbohydrates. So, less of roti, especially wheat based roti, गेहूं का आटा वाली रोटी नहीं, but more of jawar, bajra, ragi, millets based, less white rice, more of legumes, pulses, beans. So increase your protein intake, plant based protein intake. Animal based protein, there's plenty of protein in animals. But some people maybe are not able to digest those animal proteins because some people find it too heavy, or maybe because of the humidity and the high temperatures in India, it's difficult for some people to eat too much non-vegetarian food. So there are also vegetarian food sources where you can have a balanced diet and you can have high protein diet in that as well. So diabetes management, but exercise and good diet is very important for diabetes management. If you don't exercise and you just keep eating, like we have delicious food in India every day, every week, every month, and there is some festival, so we always are, eating sweet treats and snacks, and this is what is making us unhealthy because we don't like to exercise, we like to eat, and we like to rest. So that's why we expand sideways. um, but anyways, Maham, I think we can talk about that later. Let's continue with what we have to speak about. And then we have all of the things that I already told you about. And here, what I want to tell you one thing in terms of career prospects. So, the last part, driving technological innovation. So, I would suggest all of you, if you are not computer savvy, please try to get some computational courses, get some hands on computation in silico work done, especially in this day and age of AI. You need some programming, Python programming, maybe not proper coding, but at least the sense of Python programming, some Unix based systems, so Unix tools. Instead of Windows, you could go work on Ubuntu system on your PC. It's easily installable. So learn these things. There's lots of tutorials available on YouTube. Do these things. Get some bioinformatics courses. There's also a lot of YouTube resources for bioinformatics. Because what's happening now, there's a lot of data coming from protein structure. There's a lot of cryo EM structures being made, structures being solved, and all of this data needs to be processed. So companies, especially biopharma companies, are always looking for scientists who can use this data and make some good use of it, and process it such a way that they can get these structures solved, they can get these structures refined. Right, when you look at when you go to Uniprot, or when you go to what do you call, PDB, Protein Data Bank, pdb.org, you have these X-ray crystallography solved structures, or NMR solved structures. The resolution, the lower the number, the better the structure. The higher the resolution means the lower number. So if it's 1.2 angstrom or 2 angstrom, the crystal structure or the protein structure is amazing, is more accurate, is more clear. I mean, not more clear, is clearer than the higher numbers. So this thing I'm suggesting, and please take this seriously, because you will have a lot of options open for you to take up jobs, and you could also do this remotely without actually requiring you to be on site, because nowadays you can sit at home, do your work, especially when it's computational biology, bioinformatics, and protein modeling and analysis. There's also something called as what do you call, molecular dynamics simulations, MDS. So where you take your protein molecule and you create a virtual environment of, let's say, a buffer or a formulation, wherein you try to see if the protein is interacting very well with the surrounding environment, and what is the energy number of the protein. Means, is the protein fine or is the protein struggling to stay in solution? So all of these molecular dynamic simulations, they take a lot of computational power. So for that, you need high computing cluster, which means they have supercomputer clusters where you can take a cluster, or your company will give you a user ID and your own space in the cluster, and you can run these molecular dynamic simulations. Because if you try to run this on your laptop or on your PC at home, you will cook your laptop, you will cook your PC, because they will consume a lot of energy. Your PC will get heated up and you will not be able to finish the job. So it's very energy hungry. So these things, if you learn, you will become an expert. And all of the resources, I think somebody asked me Coursera and somebody asked me some. Yes, everything is fine, but companies need real expertise. Means you have to try and learn it, but also there are on GitHub, you might have heard GITHUB. You have a lot of tools available to actually, and also some lots of data available on GitHub to actually use protein, use DNA sequences to try and mine the data in such a way that you can actually create your own project work before you go for company interviews, and you can show them your work from these databases. So it's like, as if you are creating a project for yourself and you're working on that project. So just take that as a serious project and make it so that when you go to the company and you tell them, hey, would you like to have a look? I have worked on this and I have done this. So when they see that, oh, you have actually good hands on experience. Because what happens when you learn a course, you don't really know how to apply those skills. When a question is asked, hey, can you do this? And you're like, oh, actually, I don't know, but I had done it this way. I don't know how to use it. So that's where the confusion comes. But when you actually take these questions and make your own, exercise, then you can work on it. So you can take help of your friends, somebody who is good in computers. You can take help with your engineering friends. They can also teach you or give you the resources for, Unix, where you can download the software and work, because I think it's free. Unix is free. Ubuntu system is free. And yeah, Python is also free. So you can just spend some time every day, maybe 1 hour or half an hour, learn Python, and then see how you can move ahead. So, so now again, importance of protein. So that I just put out some pictures. Diagnostics, therapeutics, drug targets, biomarkers, nutritional therapy. So all of these. So I was talking about liver enzymes. I was also talking about cardiac enzymes and some antibody levels for diagnosing infections. C-reactive protein, right? CRP is a marker for inflammation. So CRP is also a protein, C-reactive protein. That is also used. So you see, proteins are everywhere, especially in healthcare. There's nothing without proteins. So no life and no disease therapy without proteins. So now recombinant proteins. So what are recombinant proteins? Now we know what proteins are, but what are recombinant proteins? So recombinant proteins are specially engineered proteins which are produced by introducing a foreign gene into a host cell using recombinant DNA technology. So Sabina, I will tell you later, but actually I would say most of these platforms who are like trying to sell, hey, do the course, this is amazing, this is amazing. I feel YouTube is the best resource. I don't think there's anyone teaching you so amazingly well that you can say, hey, if you do this course, you will be amazing in Python programming. If you give time, YouTube is free. Free, absolutely. It's your time and your internet that will be used. No money will go. I mean, money will go because you're using the internet, so you're paying for your internet pack, but it is free. So don't get into this thing, oh, I'll get a certification if I show this certification. People don't care about certifications. Unless you have a degree in bioinformatics, where you really did bioinformatics, all the 3 years or whatever, 2 years of your master's, then it's different. But if you are doing these private certifications, it looks good on the CV, but to be honest, companies really want people who know how to use these programs, how to use these systems. So I would say get on YouTube or get on any of the resources which are cheap or free or somewhere where you don't have to pay too much, because there are also people who are trying to make money off you. So you should not get influenced by, hey, this person is doing amazing course. It might be looking great, but actually doesn't make sense to spend so much money. If I were you, I would not spend too much money on getting courses from particular places. So I hope that answers your question. So recombinant proteins um, expresses the gene. The host cell machinery expresses the gene. So basically, you might have heard, or you have already experienced, possibly some of you, maybe not all of you. Some people have already done molecular cloning in their lab, or at least did something when somebody was doing cloning. At least did a restriction digestion to run a plasmid on an agarose gel. So plasmids are circular DNA, and then when you linearize them by snipping, by cutting it with a restriction enzyme, they become linear. So then it's easy to see when they run on an agarose gel, because if the plasmid is circular, it runs at a different speed. It doesn't run according to the size. So when you run a DNA marker or a ladder and you run your plasmid, which is, let's say, 4.5 kb, if it's circular, it will not run at 4.5 kb when you match it with the DNA ladder. It will run maybe higher or maybe faster. It depends. So you always have to at least cut it with one restriction enzyme, one known restriction enzyme, which will cut it in one place and linearize it and open it up. So when it's open, it will be running according to the size. So then you will be able to identify whether my plasmid DNA is actually 4.5 or something else. So cell-based production systems. So now, since you come from a microbiology background, or at least everyone has a microbiology knowledge, bacteria, very fast, very easy, cost effective, simple, non glycosylated proteins. So one thing to remember, if you are looking to produce a protein which has glycosylation, and glycosylation has a very important part to play in a protein's function. So a protein functions or folds properly, sometimes, not sometimes, but to a certain extent because of the glycosylation. So if a protein, like a mammalian protein, let's say like a hemoglobin molecule, it is glycosylated originally because it's a mammalian. Hemoglobin is not a bacterial protein, but you can still produce it. However, you will have to supply the heme, the iron, because bacteria will not have the iron. You will have to give it iron. So, you know, it will not be the exact hemoglobin that you want. It will be some globin without the proper heme and without the glycosylation. So that's why bacterial systems are not good for glycosylated proteins, but they are very good for other enzymes, other industrial enzymes, other food-based enzymes, whatever you need to do for processing your, industrial things, and even for bioremediation, for remediating your water sources by putting some, enzymes so that the biomass, the bioburden of the water reduces. And then people do COD, BOD to test if the water's COD, BOD has reduced after these enzymatic treatments. So when you have these wastewater treatment plants, they also do some chemical as well as enzymatic treatments. So that's where bacterial proteins, which are produced in bulk and cheaply, that comes in very handy, very useful resource. And then you also have cyanobacteria. So the cyanobacteria also, just like E. coli, are prokaryotic, but they are photosynthetic microbes. They are also simple and cheap. So some proteins are already being grown in cyanobacteria, and you just need light and you need some medium, and they are absolutely fine. So you don't need to really take care of them, just like how, um, not just like, unlike mammalian cells. Mammalian cells are very expensive. One mammalian cell culture, 1 liter bottle costs, I don't know, 5,000 rupees at least. So you can imagine, if you waste 1 liter of bottle, 5,000 to 10,000 rupees gone for waste. And the cells, they come in very small vials. You have to thaw them from -80. They are shipped from America or Europe. And these cells also cost thousands, multiple thousands of rupees. So, cell culture, mammalian cell culture, is a very expensive work and a very expensive business. But if you can get your protein from algae, from yeast, or even insect cells, and get it to fold and function properly, nothing like it. But for certain vaccines and certain types of, molecules like antibodies, you need to use the mammalian cells. So Big industries, biopharma industries use mammalian cells, mainly CHO. A HEC is used for transient transfections. So I will talk more about it later. I'll just finish this. And the system choice depends on protein characteristics. What kind of proteins you want? Like you want glycosylated, don't go for E. coli. You can go for yeast, insect cells, mammalian cells, and also algae, microalgae. But depends what kind of glycosylation, because a particular type of glycosylation makes what do you call the function possible. If the glycosylation is not correct, if it's another glycosylation, then the protein might not function as effectively as it should. And then, just to give you an example here, I wanted to give you a mixture how somebody, some scientists, I have given the source below, scientists in Russia. So they are using plant membrane proteins. They are removing the membrane of the leaf, and they're getting the DNA out, and the gene of interest is being isolated. So your DNA source is from the plant, and that is then put into a vector, which is a plasmid vector, a cloning vector, and then you transform it into E. coli and you plate it. After you plate it, and then you take the colonies, you again like kind of culture it, and then you extract out the plasmid. You know plasmid mini prep, right? You must have done plasmid mini prep or Qiagen kit, QIAGEN, Qiagen plasmid mini prep. Maybe you would have extracted plasmid DNA by setting up overnight cultures of E. coli by putting, a DNA or a toothpick or what do you call the loop, wiggle it in the bacterial cell culture LB media, and then next day you are able to, um, extract your plasmid DNA. So I suppose you might have done. If you haven't, then this is a very, basic information for plasmid extraction, and that is then uh, transformed into yeast. So now you confirm that the vector has been created. It's now showing colonies on E. coli. Now we'll make more of this DNA. And that DNA is then transfected into yeast. So did you see what happened? Plant gene goes into E. coli. E. coli gene then goes into yeast. Why? Because plant cells are also glycosylated. Now, the E. coli was just a medium for the DNA to be made. But actual work of production of the protein is going on in yeast, because the yeast protein product will be sort of similar to the plant based product. So the functional assay will be possible and will be biologically relevant when these two systems are compared, not when you produce it in E. coli. So that's why I wanted to bring this, that you have to remember what is the reason and what is the end goal of your protein. If you just produce it for the fun of it, fine, but it has to serve a purpose. What is the purpose? That is very important. Before you do the protein production, before you do the cloning, always think it through, the end goal, and then you will be able to plan your experiment better, and hopefully you will get better results. And now, one very interesting thing, something that I use on a day to day basis, because I have done bacteria, I have done mammalian cells, I have not done algae, I have not done cyanobacteria, I have not done yeast, I have not done insect cells, but I have produced protein in bacteria, in mammalian, and in cell-free system. So from the last 1 year and 1 month, I have been in this company, LenioBio, where we produce proteins without any cells. We actually take the protein manufacturing machinery from plant-based system, which are tobacco cells. So tobacco leaves cells, they are grown in bioreactors, and then we harvest these cells and we remove whatever is inside the cells, and that includes the ribosomes, the mitochondria, and all of the cytosolic material that is inside the cells. And that's how a lysate, a yellow colored lysate, which you will see in the pictures later, to which we add DNA of our choice of the protein that we want to make. And we are able to make protein just in a matter of 1 to 2 days. So that is proper, really fast system. And that was one of the reasons why I wanted to know how we do it. That's why I joined this company. And now I'm making proteins that are difficult to express, because when you are expressing proteins in a cell based system, sometimes proteins are also toxic to the cells. You will read some papers. If you see some papers online, just type on Google toxic proteins for cells. You will see a number of proteins where scientists failed. Bacteria died, yeast died, mammalian cells died. When the protein was expressed, after it reached a critical level, the cells just died. Why? Because the protein had some kind of a functional feature which was able to lyse the cells on the inside. It was trying to eat up the cells. So that turned out to be toxic. So we have cell-free system. Why? Because now with this cell-free system, we are able to produce difficult to express proteins from other systems. The other systems that cannot produce, we produce it. And we produce proteins which are more than 2,000, 3,000 amino acids long, which cannot be produced in E. coli, which cannot be produced in mammalian cells. We produced it, and for gene therapy, we produce some proteins which was not possible from any other systems. That's why that company came to this company where I'm working. Hey, can you please make it for us? Because we needed to use it for a gene therapy project. And we tried. It was difficult, but we somehow managed. So that was a good success story. So now types of proteins. So just to give you an overview, so these are TPRs, these are repeat proteins. So as you see, they are just like, spiral, 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 like ribbons, twisted, 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 like a right-handed alpha helix of a DNA. So these are kind of twisted. And so I have shown them in the ribbon form, the cartoon form. So A is from Shigella species. It is a dimeric protein. It's from Shigella, Shigella, Salmonella. So Shigella species, gram-negative bacteria. The B is from Yersinia enterocolitica, like I would say a cousin of Yersinia pestis, the one which causes plague. So I worked on the protein which is in B, and I did the biophysical characterization of this protein. So that was my PhD project actually. Maybe you might have heard type 3 secretion system. There is type 1, type 2, type 3, type 4, type 5, type 6, and up to type 7 or 8. So gram negative bacteria, they infect using these secretion systems. So this protein, and then the third one, the C1, is from Pseudomonas aeruginosa, which causes cystic fibrosis. So yeah, so these proteins are also very similar, and these are TPRs, tetratricopeptide repeats. Now, can you tell me, but looking at this protein structure, what do you think uh, would it function as? What would be its function? If you see something like this, jumbled up and twisty, how would it function? What would it do? Can you just guess? Any of you? You can write it in the chat. Anybody?
Sir, they may have hydrophobic regions and they can be act as a transmembrane protein or something like that.
Mm, partially correct. They have some hydrophobic, but they, let's say, have you heard of chaperone?
Yes, sir. They help in
Yes.
protein folding and to
Yes.
maintain their growth safe.
Yes. So, so from that you get a clue that these proteins help other proteins. So what happens is the structure of these proteins is such that they have these grooves and helices. So when you have the grooves twisted, you have other proteins that come and get into the groove, and they kind of get pulled towards this protein. And then another protein gets pulled towards this protein. And because that protein was already pulled and this protein also got pulled, these two proteins now can interact with each other. So this acts as a scaffold. Scaffold, when buildings are being built, construction workers, build these bamboo staircases to climb on top. So they make scaffolds so that they can actually go on top of buildings. So these proteins are also similar to those bamboo scaffolds, like bamboo ki seedhi, so that other proteins can come and climb or come and rest. So these proteins have a very important role in protein protein interaction in our cellular signaling mechanisms. So have you heard anaphase, metaphase?
Yes, sir.
Yes. So have you heard of anaphase promoting complex APC?
Yes, sir.
So these proteins actually form your anaphase promoting complex.
Okay, sir.
So, I mean, I had inserted another picture, but I think it went away somewhere. But doesn't matter. I wanted to show you a different protein, but maybe I can just tell you. You know, right? What a beta sheet rich protein looks like. What beta sheets look like. Alpha helical, beta sheet rich proteins. So these are alpha helical, and then there are also beta sheet proteins, and then there are also coiled domains in proteins. So these are the different types of architectures of proteins, and there are different protein families where certain proteins have certain types of structures, and that's why they perform certain functions. And then you have globular proteins like hemoglobin, which have a certain function. And then you also have albumin, which also performs a certain function. So these are globular proteins, whereas these are repeat proteins. Repeat proteins are scaffolds. Globular proteins have a typical function, a specific function. Like enzymes, for example, they are also globular. So, but enzymes, remember, they have a reaction center. And what happens in the reaction center? Which ion is important for enzymatic activity? Can anyone tell me? Which elements are important for enzymatic function apart from the protein itself? Can you
There
name one?
there are different types of cofactors, like
Yes.
for some enzyme, magnesium, or for some enzyme, zinc act as a cofactor.
Yes. Yes. Magnesium. You said it very correctly. Yes. Yeah. Also, purple and mega. Everyone is correct. So you got it right. So magnesium is very important. Yes. So in the active site, the ion goes and docks, and when it docks into the active site, that's when the enzyme is able to work on the substrate and produce the product. Without the magnesium, the enzyme will remain inactive. That's why whenever you see enzyme vials, commercial enzyme vials are given and they are available, they actually give you um, a formulation. So the formulation of whatever buffers, and they also say 5 millimolar MgCl2 or 50 millimolar MgCl2. Why? Because magnesium is coming from the magnesium chloride. So that you have to remember. I can explain that. So beta sheets are basically these are flat pleated sheets, which more like now these proteins, alpha helical proteins, they have a very nearby interaction. So this interacts with this, interacts with this, interacts with this. But beta sheet proteins, what happens is these proteins have flatter sheets, and they can interact not just here, but they can also interact from position number 20 to position number 108. So these proteins have short range interactions. Beta sheets have long range interactions. So that's why in globular proteins, you have beta sheets and some alpha helical. So there, the interaction of C terminus is also going to happen with the N terminus. But in this case, N terminus is very distinct and away from the C terminus. So do you understand what I'm saying? So N and C are far away. But in a globular protein where you have also beta sheets, the N and C might be very close to each other, next to each other. So that's how this interaction happens. So have you heard of amyloid proteins? Amyloid? Yes. So amyloids are beta sheet proteins. They are typical example of beta sheet rich proteins. So they, prions, prions, prion disease.
Yes, sir.
Yes. So prion disease also has beta sheet proteins. So now coming to the cloning part, how many of you know how to make PCR primers? Have you made PCR primers? Okay. Okay, so if you have not made PCR primers, so let me just tell you. So first, you take a look at your gene sequence. Now, can you still see my screen if I open this window? Can you or can you not? If I change my window, can you see it?
No, sir.
Okay, so I will just switch to a different window. I just want to show you how to wait, select a